Hi BioNerd, this is Modern Chicken channel where you can expand your information about all branches of biology. But before we start, please don't forget to subscribe and follow me on my Instagram. So previously we talked about the structure of the neuron. Now we'll move to the glial cells. There are 4 types of glial cells: the astrocyte, the ependymal cells, the microglia, and the oligodendrocyte. We'll start talking about the astrocyte. Well, when you have the prefix astro, that means the thing that you're describing is similar to a star. And that explains why the astrocyte is just a glial cell that has the shape of a star. Well, the astrocytes have multiple functions, but the most important one is making junctions between the cells that form the capillary inside the CNS, and then making what we call the BBB, the blood brain barrier, which is considered a highly selective filter to the substances than those presented between the blood and most other tissues. Then we have the second function, which is potassium ions storing. And here we want you really to focus, because I'm going to explain something important. Here you have a cell in the CNS, and here you have the extracellular fluid, and here comes our hero, the astrocyte. So usually you must just keep in mind this information. The concentration of potassium ions inside the cell is higher than the concentration of the ions outside the cells. Potassium ions, I'm talking specifically about the potassium ions. Other ions like maybe sodium are more concentrated outside the cell than inside, but we don't want that for now. All what we care about now is potassium. So usually the substance tends to move from the place where its concentration is high to the place where its concentration is low. And that's why the leakage channels of potassium will let the potassium move from inside to outside the cell. Do you think it's just a random operation or a random thing that we don't need? No, it's a very important process that we need daily. But if just potassium continued to flow from inside to outside the cell, wouldn't the extracellular fluid achieve the balance? So the inside of cell would be exactly like outside the cell, and that's why the potassium channel would be closed, and then we'll have accumulated potassium in the extracellular fluid, and which is really, really bad. We don't want that to happen. So what must we do? We must just let this potassium gets out, and when it's accumulated, you can just store it in our hero, the astrocyte. So the astrocytes will take this additional potassium and will prevent it from being accumulated in the extracellular fluid. The second or the third function, I think, is giving glucose and getting rid of the metabolic waste product like ammonia. And then we have the last function we'll talk about. Actually, there are much more functions, but those are the basic ones that you need to know. The guiding, the neurons guiding. So during during embryonic development, you'll have your neurons in the CNS guided to their destination properly by the astrocyte. So it's a very important kind of glial cells. You must memorize it. You must know more about astrocytes if you want, but basically that's all that we need. Then we have the second type of the astrocyte of the glial cells, which is the ependymal cells. Well, I don't really have lots of things to say about ependymal cells, but I really can say that they line, as we can see here. Here is an ependymal cell, and here are just all the ependymal cells. So they line the fluid-filled cavities inside the spinal cord and the brain, and they also regulate the production of the corticospinal fluid, which will be described later. The corticospinal fluid. And lining the fluid filled cavities. That's all what we need to know about ependymal cells right now. And then we have a third type of glial cells, which is this little thing, the microglia. So you think it's just a tiny thing, but it's really a very important one, because it's responsible of the immune responses inside the CNS, and it's also responsible of phagocytosis, which is the engulfing and digestion of the harmful materials. It also plays a role in synapses remodeling, and that will lead to what we call plasticity. If you don't know what is plasticity, it's the differences in synapses, and it's the remodeling of your synapses during your whole life. And that's why you are, as a child, not the same as you as a person, as an adult person, maybe a teenager. That's why me, maybe as someone that doesn't know anything about German language, differ a lot from you. Maybe you are an experienced, or you know lots of information about German language. So we really differ at different fields and at different ages, and plasticity can explain all of that. We're really gonna explain more about plasticities after talking about action potential, after talking about consciousness. We really have a series of in neurology which will be very, very interesting. But for now, all what you need to know that microglia is responsible of immune responses, and for plasticity and for phagocytosis. And now we'll move to the fourth type of glial cells, which is the oligodendrocytes. So usually when you have the prefix of oligo, that means a little, or you don't have lots of the thing that comes after it. Dendrocytes come from dendrites, the dendrites that we explained about previously. So it's just a glial cell that doesn't have lots of dendrites. That's the explanation of it. And it has an important function, which is making myelin. Now, the neurons or the axons of most neurons are covered by a sheet called myelin sheet, which consists of 20 to 200 layers of highly modified plasma membranes that are covered, or that wrap the axon by a nearby supporting cell. So these nearby supporting cells in CNS, and only in the CNS, are the oligodendrites, while in the PNS, the peripheral nervous system, we have what we call Schwann cells. And now we're going to compare between oligodendrites and Schwann cells. Oligodendrocyte Schwann cells. First of all, Schwann cells are found in the PNS, while oligodendrites are found inside the CNS. The second thing is that one oligodendrite produce myelin for as many as 40 axons at the same time. That's what we can see here. Here you can find only one oligodendrite, and here the axon is the myelin is distributed to multiple axons of multiple neurons. So it can produce myelin for more than an axon. But here in Schwann cells, one Schwann cell can produce myelin to 1.5 segment of the axon. So it will be just like this. Along the axons, you'll have regular intervals along the axon, like this, until you reach the terminals. So they really differ, because here one cell means one segment, while here one cell produces myelin for as many as 40 axons. The third thing, which is very, very important, I want really you to focus here: oligodendrites, if they are damaged by any case, if something happened and now they are damaged, they can't be regenerated. They usually can't be regenerated at all, and it's very hard to get them back. However, when you damage Schwann cells, it's easy to regenerate, or it's not easy, but at least it's possible. Yes. No. So there's something that you must know. As we talked previously, the nervous system doesn't have a centrosome, and that explains why it doesn't divide. The neurons, either they are inside the CNS or in the PNS, can divide. But the difference is that glial cells, not the neurons, only glial cells can really divide. But specifically oligodendrites can't be regenerated at all. And to make something regenerated, or to make a neuron regenerated, you have two things that you must achieve. So the neuron must be in the PNS first, and second, the neuron really must be, or the damage must be, in the axon. So if the body is damaged, there is no hope to make the body be regenerated again. But if an axon in the PNS is regenerated, it can be regenerated. And if any neuron in the CNS is damaged, there is no hope at all. Actually, it's a very little hope to make them regenerate again. And none of them can divide to produce new things at all. But they can, at least in the PNS, regenerate. But in the CNS, they can't neither divide nor regenerate. But the glial cells can actually divide and can regenerate, but oligodendrites cannot. That's all. We talked about the structure of the neurons. We really have a trip to talk about the principles of neurology. In the next video, maybe we'll talk about plasticity or consciousness, maybe about action potential. I'll decide about that later, but you'll have a very interesting series of neurology. Thanks for watching.